dear students. I hope you are doing well. Today we are going to discuss about DNA uh, the, uh, and the chromosome. And this is the topic is coming from my lecture series. And in that we are going to discuss about different type of biomolecules. And this is a part of the basic concept of genetics. So this is the journal science. Uh, this is the means course of general science, and this is my lecture number 8. So in this lecture we are going to discuss about that what is the structure of DNA, how the DNA organize and making the chromosome. And okay, so going to start from here. So first of all we are going to discuss about what is DNA and how it is looking like and what is the structure of DNA. So DNA, as its name is showing, that DNA is a deoxyribonucleic acid. That is its long name, and short form we call it like a DNA, deoxyribonucleic acid. So DNA is actually is a polymer of nucleotide, and DNA is a biomolecule. DNA is a macromolecule. So why DNA is a biomolecule? Because it is having a biological activity in our cell. And why it is a macromolecule? Because it is composed of many small um, molecules, and that we say it like um, many small uh, um, many small of the pieces of molecule. And DNA composed of when we talk about the structure of DNA, we can see here DNA is one molecule of DNA is consist on two DNA helix that are coiled on each other. And if I'm talking about the structure of DNA, so we can see here this DNA is composed on nucleotide, and then each nucleotide is consist on a sugar base and a nucleotide and a sugar base, a sugar moiety, and that is a deoxyribose, and then a purine or pyrimidine base, and then a phosphate group. So this is the one nucleotide. One nucleotide composed of a um, sugar moiety, a purine or pyrimidine base, or a phosphate group. So on the basis of number of this phosphate group, we can say it a dia, tria, or mononucleotide. If here it is only one phosphate, so we can say it's a mononucleotide. So for example, in case of ATP, ATP is adenosine triphosphate. So it means in adenosine ATP, this is a nucleotide in that we are having three phosphate. So in another lecture that would be exclusive with the only structure of DNA, we will be discussing more about the structure of nucleotide and how these nucleotide are formed. But for this lecture, because this lecture is based on the DNA and its organization, how DNA organize on itself and making a chromosome. So here we are just thoroughly discuss about the structure of DNA. So from this in this lecture, we have to just keep in our mind that DNA is a double helix. DNA is a polymer of the nucleotide, and it is one DNA molecule is composed on two helix that are coiled on each other, and each helix is composed of several many thousand or many or several nucleotides that are joined with each other via phosphodiester bond. And how these two helix join with each other? These two helix join with each other by the hydrogen bonding that are present between the pyrimidine or purine nucleotide. So this is how the two helices join with each other. So here we can see is a molecule of DNA. Now from this molecule of DNA, that is very initial level of organization, we will be seeing that how this simplest molecule of DNA that is composed of consist on two helix of uh, uh, two DNA helix organized on itself and making a chromosome. So this single molecule, what happened in the second level of second organizational level, that this single helix of DNA coiled with a protein that we say it's a histone and making a molecule or making a complex structure that we called is a nucleosome. And this is the second level of organization when the this double helix coiled around the protein uh, uh, protein that we say is a histone. So this histone is a octamer. We will be seeing in coming slide that what protein subunit are present in this octamer. But here we just have to keep in our mind that in second level of organization, the double helix of DNA coiled around a protein histone and making a molecule or making a means complex that we say is a nucleosome. And in the third level, what will happen? The one what will happen that again this DNA double helix again coil on the this histone, and once 1.65 time of this DNA helix would be coiling around the nucleosome. So this would be the third level of third organizational level of means means organization or organization level of the uh, means how DNA organize. So in the third level, what happened? We will be having a histone octamer that would be found to coil with 1.65 time of the DNA helix around it. So in the fourth level, what happened after that? One another histone protein that we say is a H1 protein, H1 histone would be binding over this nucleosome. So what is the purpose of binding this histone, H1 histone, is that so the DNA that is already means um, coiled around the nucleosome could not be means unwrapped. So this is the fourth level of organization. And then once this H1 stone bind with this nucleosome, we say it's a chromatosome. So in the fourth level, we will say it's a chromatosome. Then what happened? This chromatosome will be coiling many hundred means chromatosome or many thousand of chromosome um, coiling with each other and making this 30 nanometer fiber. And then what happened? This 30 nanometer fiber coil on each other in this way and making 300 nanometer loops. And then what happened? This 300 nanometer loop will be coiling on each other and making 250 nanometer white fiber. And then what happened? This 250 nanometer white fiber making this 700 nanometer um, seven means making this 700 nanometer fiber. And then what happened? They will be keep coiling on each other, and and then they eventually making a very complex structure that we say is a chromosome. So chromosome width is 1400 nanometer, means this coil 700 nanometer, this 250 nanometer fiber keep coiling on each other until and unless we will be getting 1400 nanometer structure that we say it's a chromosome. So we see here that how the DNA molecule coiling with the protein and making the chromosome. So we just have to keep in mind few terminologies here. One is a double helix DNA and other is a nucleosome. What is a nucleosome? When the DNA double helix coil around the histone protein. And what is a chromatosome? When the H1 histone attach with the nucleosome, that would be the chromatosome. So here we can see that this is a we are having a chromosome, and now if I am talking that what is a chromosome, actually chromosome is a single length of DNA that is wrapped many times around lots of protein that we say is a chromosome. So now we are going to talk about the what protein are present in this chromosome. So the chromosome is having protein that we say is a histone protein, and histone protein is a octamer. It is having two subunit or two molecule of H2A, histone 2A, and two molecule or two subunit of histone 2B, and two subunit of histone H3 and two subunit of histone H4. So that is how it is a histone protein that is present in chromosome, and it is a octamer because it is having four different type of histone protein that are present um, uh, in a couple. So, uh, so when I am going to talk about again that what is a chromosome, chromosome is a structure in that um, a histone protein is found to be coiled with the DNA molecule or DNA double helix. So, little bit we are going to discuss about the structure of the chromosome. So, chromosome we can see here it is having two arm. Short arm we say is a P arm, and then a long arm is called as a Q arm, and then in the middle of this structure we say it's a centromere that is joining two chromosome pair of chromosome with each other, and then very end we find it is a telomere, and this telomere actually protecting this chromosome from the breakage. So in any other lecture we can more discuss about the what is the telomere and what is the more means as comprehensive structure of the chromosome. So now we are going to talk about the gene. What is a gene? So we understand that what is a chromosome. So what is a gene? Gene is actually a specific segment of DNA that is containing instruction to make the protein. So we see here that in the cell we find the chromosome, and chromosome is consist of a DNA and the protein, and we see the single means molecule of DNA here. And what is a gene? Gene is a segment of the DNA um, uh, that is having some instruction to make a particular protein. So, and and how these instructions are present in the DNA, or this instruction is present in the DNA with the different repetitive pattern of the nucleotide that we say is a codon. So we will be discussing in coming slide. So, but here we have to understand that what is the difference between the chromosome and a gene. So, if I am talking about, we can see here two picture. One is a structure of gene and one is the structure of chromosome. So, what is a gene and what is a chromosome? Gene, it's only a molecule of a DNA that is having some important nucleotide or and these nucleotide give us the information of the type of the protein that is going to be made by the cell. So we can say that gene is a stretch of DNA that is having some information. And what is a chromosome? Chromosome is having DNA coiled with a protein histone. So what is the difference between gene and the chromosome? Gene is not having uh, protein, but the chromosome is having protein and the DNA. Gene is also only having nucleotide. Gene is having only nucleotide that are, and the some specific repetition of nucleotide is having some important information to make the protein. However, chromosome is having both DNA and the protein. So this is the main difference between DNA and the chromosome. So now we are going to talk about that how many chromosome we are having in our cell. So male and female having different set of DNA. So why they are different? The different difference in the last set, that is the sex chromosome. So we are seeing here a karyotype or a snapshot of all complete set of chromosome that are present in the female cell. So all male and the female are having um, uh, a diploid means male and female cell are having 23 pair of chromosome. So if we are talking about the female cell, so female cell are having um, a female cell means the autosome that are not the sex cell. So the means sex cell means they are not the sperm or the ovum or the egg cell. So the autosome, female autosome, or having female autosome having 23 pair of chromosome, and um, 23 pair of chromosome, and then female. Okay, okay, where is the, okay, so female cell, all female cell is having all male and female cell having 23 pair of chromosome, and in that the 22 pair of chromosome in both male and female are autosome, and the last set of chromosome is a sex chromosome. So in the female, these sex chromosome are XX. Both chromosome are X in the female. And the if we are talking about the male, male are having um, 22 autosome, same 22 chromosome as a autosome, and the one set of sex chromosome, in that they are having one X and one Y. So what is the difference between man and woman chromosome set? The difference is the sex chromosome. In the female we are having 2X, and in the male we are having 1X and 1Y. So this is the Y chromosome that is define the sex of the baby. So now we are going to discuss about the genetic code. What is a genetic code? So we know about that. What is a gene? A gene is a stretch of the DNA that is having repetition of the nucleotide, and these repetition is having some information to make the protein. So this is the segment of a DNA, and what is a this is a segment of RNA, and what is a genetic code? Genetic code is a codon that are present either on DNA or on the RNA, and this is the that is consist on a three nucleotide. So um, a trinucleotide is a codon. So we are having a stretch of nucleotide here, that is RNA, and the three nucleotide in a on a segment of DNA is called as a codon. So what is the difference between DNA and RNA? The difference between DNA and RNA is the difference of this nucleotide, that is uracil. In RNA we are having uracil, in DNA we are having adenine, thiamine in place of this uracil. And what is a genetic code? Genetic code is a sequence, or what is a code? A codon. A codon is a three nucleotide, making up by the three nucleotide. So we are having different start and stop codon on the DNA. So AUG, GUG or UUG, this is the start codon. What is the start and stop codon? We can understand when we will be understanding about the protein synthesis. This start and stop codon allow the protein synthesis to be start or the stop on different time points. And what is the stop codon? Stop codon, normally if we are finding in the stretch of nucleotide in DNA and RNA, UAG, in RNA, UAG, UGA, these are the stop codon. Stop codon. So UAG we normally say it's amber, and UGA we say is a opal. So this was all about the today's lecture. In today's lecture, we understand that what is the structure of DNA, how DNA coil on itself and making the chromosome, and what is the structure of the histone protein, what is the structure of the chromosome, and what is a gene, and what is the difference between chromosome and gene, and what is the difference between the chromosome in the male and the female. And we also understand about what is a genetic code and what is a codon, and what is the start codon, what is the stop codon. So keep watching my video. Thank you very much. Allah Hafiz.